Hello everyone. What we're going to discuss in this lecture is a second way in which some type of cells can divide differently from what we discussed in the lecture talking about the cell cycle and the division of the genetic material by mitosis, where if you remember, you have a diploid cell that gives origin to two diploid cells, so two identical cells through mitosis. In this case, what we're going to be talking about here is how some cells, germ cells, that are diploid give origin to actually four cells with a single set of genetic material. So all haploid type of cells, which are basically the cells that are involved in the process of fertilization later on. One important thing I want you to take home from this lecture is also how this process of meiosis ultimately leads to the generation of cells that are all different from each other, and how in the end, through the process of fertilization after that, all individuals generated by these cells are going to be genetically basically different. So there is not a single individual among the human population except for identical twins that do share the genetic material. Not even with your brother, you have basically no chance, or brother or sister, to have the same genetic material based on all this mechanism of variability that we'll talk about here in this lecture. Obviously, variability is very important for living organism. You see when we talk in BIO359 about evolution, you see how, for example, natural selection works by selecting organisms because within the same population or within the same species, there are individuals that are genetically different. So different organisms, they deal differently to produce this type of genetic variability. In general, individual based on the based on the way they reproduce can be divided between asexual and sexual organism. A sexual organism, they give origin to the next generation simply by dividing. They don't mix genetic material. Bacteria are an example, so prokaryotes in general. All they do, if you remember when we talked about the prokaryotic cell cycle, all they do, they duplicate their DNA and then they generate two cells with basically identical copies of the same DNA. In this case, obviously, their variation can only depend on mutations that happen through replication of the DNA or through external elements like UV light and so on. Obviously, to deal with that, to be successful, they deal with mutations, but also high numbers. By producing high numbers of individuals, like billions of bacteria are produced by a single bacteria in a few hours, by working with numbers and mutation, you can create enough variability to be successful throughout evolution. Other organisms, instead, they reproduce sexually. So you have two different organisms now. They produce certain cells called gametes, which have half of the DNA. They mix it together. By doing that, through the process of meiosis as well, which we'll talk about later on, they can actually produce extreme variability even with small numbers. So even by having a few offsprings, which obviously they have to be protected until they're successful to reproduce again, even with few offsprings, they have enough variability within the population to guarantee to be successful enough throughout evolution. And we'll see the mechanism. You see, when we talk about meiosis, meiosis introduces variation, producing gametes that are different from each other through mixings of DNA, which is referred to as genetic recombination by crossing over, and by basically randomly assigning the chromosome present in the original cells into the different gametes, which is referred to as independent assortment. And fertilization as well, by choosing randomly the different gametes that are going to fuse together to produce the offspring, contributes to this variability as well. So variability in a sexual individual is achieved through mutations only, and then large number help with that. In sexual individual is achieved through the process of meiosis and fertilization done with two different individuals mixing the DNA together. So from now on, we'll focus only on sexual reproduction. Okay, so this gives origin to reproduction, as we said, of gametes, which are produced by meiosis. This is a special type of cell division, okay, which we'll see in a few minutes. Summarizing key points here. So meiosis leads to the production of gametes, which have half of the chromosome, which are present in the cell that gives origin to the gametes. So there are dedicated cells called germ cells that give origin to these gametes, and the DNA will go from double copy, so from a diploid type of cells to a haploid type of cells, which are the gametes. And then at fertilization, what you see in this schematic here, so the germ cells here of two different parents, so this will be in our case a male and a female. These germ cells in the gonads, so it will be testis for men and in the ovaries for a female. These germ cells, they do produce gametes through meiosis, which have only half of the genetic material. And then, through the process of fertilization, when you have a sperm and an egg joined together, now you have the production of first a single cell called zygote, which then will give origin to the next generation, the next individual, which will be diploid again as the original parents. Now, both meiosis and fertilization, they lead to mixings of genetic information, as we say before. And this basically leads, if you think about it, both in an individual like yourself, you have some sort of cycle there where you have that as a diploid organism, some of your cells, they do pass a certain phase where they are haploid, and then they regenerate a diploid phase. So this is referred to, moving on fast here, as a life cycle. The life cycle is the cycle that all living organisms go through, or sexual living organisms, I should say, go through, where the diploid organism produce some haploid cells or organism, which then will regenerate the diploid organism, which goes back and does the same thing. Connecting basically is the replication of the individual, the cycle that leads to the replication of an individual into other individuals, which then go back to the same. Now, when you compare the life cycle of animals to the one of plants, there are big differences in this case. The cycle is the same, but the main difference is that if you look at animals, basically animals, they spend the vast majority of their time in the diploid phase. Think about yourself, for example. You are diploid. Go look in the mirror. All you're looking at is yourself. All the cells in your body, they are diploid, except for a few cells in your gonads, which you can't see like that, which have been produced by the germ cells, which are haploid. And these are the cells that later on, through fertilization, you get to join with the cells from another individual to produce another individual diploid like yourself. So your life cycle basically consists vast majority of the time in the diploid phase, and the haploid phase is just very, very limited within your organism to a few cells, okay, which will never give origin to an individual on their own, but they have to fuse together to produce another visual individual like yourself. This is different from plants, and we'll talk about this in more detail in BIO359. But just going quickly through this, for example, this is the life cycle of a fern, one of those beautiful plants that you see quite abundant in forests, particularly if you go in North Ontario. So these plants right here, the one that you used to look at is actually the diploid form. So it has two sets of chromosomes there. Then this plant, instead of having like gonads within itself like we have, what they do, they produce by meiosis, they produce some cells which have only half of the genetic material. So this is diploid, this is haploid, which actually doesn't fuse directly with other cells. First generates another organism, which is still a fern, but is different from the diploid form, which actually grows like any plants. So they actually have an external haploid phase, which takes a long time. These plants will grow and grow and grow. At a certain point, will start producing, by mitosis in this case, the gametes, so the egg and the sperm, which then will fuse and will give origin again to the diploid phase, which is the one you see. Next time you look at ferns, look carefully around it, and you might see that actually there are plants which don't think they're fern, but actually they are just a haploid form. So this just to tell you that plants in general can actually have phases, separate phases, which leads again to plants, actual real plants existing in this phase. In some plants, you'll see in BIO359, the haploid phase is actually the main phase. That's the one that takes most of the time in the life cycle. So there is a difference between the life cycle of plants and animals in terms of the haploid phase, whether it's independent, how long it lasts for, and so on. Okay, so now we'll focus on meiosis. So meiosis, again, is that process through which the DNA from a diploid cell, the germ cells, is divided, producing gametes, which are cells that do have only half of that genetic material. And this also is the process that generates the huge genetic variability found in sexual individual through two main mechanisms, which we'll repeat over and over so you take them in. The first one is the independent assortment. So the chromosome here can randomly fall into one gamete or another one. And the second thing is the exchange of genetic material through crossing over, which we'll talk about. This is an overview of meiosis. So meiosis consists of two phases. In the first phase, first of all, the DNA gets duplicated in the S phase, like in the cell cycle, if you remember, by mitosis. So DNA gets duplicated, so you get two sets now, two full sets of chromosomes there. And then through two successive steps of divisions, this genetic material here gets first divided through the first step called meiosis 1 into two nuclei right here, which then through a second step, so these two nuclei will already have only one of the two possible homologous chromosomes for each pair. If you remember, each pair of chromosomes, like chromosome 1, has two copies referred to as homologous chromosomes, one coming from your mother, one coming from your dad. Same thing for chromosome 2, 3, and so on. So at this point here, this cell would have only one of these two chromosomes, either the one coming from your mom or the one coming from your dad. So this cell will be already haploid. But they would have two sister chromatids for each of these chromosomes, which then get separated during meiosis 2 in two further nuclei here, so which then will give origin to 4 different cells, which then will have just one chromatid or chromosome here. So it will be haploid, all of them. 4 different haploid cells, which are going to be your gametes, basically. Okay, so let's look at meiosis 1. In meiosis 1, this is the most important part of meiosis, particularly prophase, the very first step in meiosis 1. So in meiosis 2, the phases have the same name that in mitosis. So you have prophase, prometaphase, metaphase, anaphase, and telophase. But these phases are referred to with a number corresponding to the meiosis you're talking about. So for the first meiosis, you have prophase 1, metaphase 1, anaphase 1, and telophase 1. And then for meiosis 2, you have prophase 2, metaphase 2, anaphase 2. So a key step is what happens in prophase 1. This is the big source of genetic variability. So what happened in this case, differently from mitosis? Try to focus on the differences between meiosis and mitosis to memorize meiosis. What happened here is that you have the first, so when the DNA is duplicated in the S phase, you have 4 different chromatids, right? 2 identical to each other and corresponding to the chromosomes, the homologous chromosomes. Now, in meiosis 1, in prophase 1, these different homologous chromosomes with the 2 chromatids each don't get separated. They don't go all over the cell, but they actually stay together almost parallel to each other, like in this picture. So you have the 2 chromatids, sister chromatids, for one of the 2 copies of the homologous chromosome here in red. Let's assume this is the one coming from your mother. The blue one is coming from your father, two identical copies here, and they stay together. This is what I refer to as a tetrads, because you have 4 chromosomes put together here. Now, during this step 2, so once this process is referred to as synapsis, the formation of the tetrad, and this is kept together by protein plus enzymes that do what they do. They actually exchange some genetic material between the 2 homologous chromosomes. So here you have one part, if red was the one from your mother, for example, one part of the chromosome from your mother has been transferred to the one from your father, and vice versa, the same part from the father has been transferred to the mother. This process is referred to as crossing over. It's a process of genetic recombination. And then after that, you see, it's still in meiosis 1, summarizing some parts here. You have that then this chromosome, they will randomly be assigned to one gamete or the other. Meiosis 2, just as an overview, in the end is nothing else than basically exactly what happens in mitosis, where in this case the sister chromatids get aligned and they get separated to 2 different poles, different nuclei. We'll go through that. So let's go through the steps of meiosis. Same naming as in mitosis: prophase, prometaphase, metaphase, anaphase, and telophase, but with the number corresponding to the specific meiosis. So prophase 1 or prophase 2, prometaphase 1, prometaphase 2, and so on. Now, so DNA has been duplicated in the S phase. The cell, the germ cell, goes through the first meiosis, so meiosis 1 first. First phase is prophase 1. In prophase 1, as I said before, so imagine these 2 being 2 homologous chromosomes, the red one coming from your mother, and this another pair, so this will be, say, chromosome 1, and the shorter one, chromosome 2, from the mother and the father. So in prophase 1, as I said before, these chromosomes, they do pair with each other, forming tetrads. Each one of them is made by 2 sister chromatids, and they basically get aligned with each other here. So tetrads are formed during prophase 1. Other than that, and we'll see what happens with that later on in the next slide, other than that, all the other events are similar to what happened in mitosis. You have the formation of a mitotic spindle with the 2 centrosomes here moving sideways to the 2 opposite poles here. And in the next step, you see that the nuclear membrane will start disappearing too. But the interesting part here, unique part, is that what happens to these tetrads in prophase 1? These tetrads, as I said before, so they start basically some parts of this tetrad. So you would have, for example, this is chromosome 1, chromosome from your mom in red, the one from your dad here, 2 sister chromatids. They are kept together by a series of proteins here, and some enzyme, what they do, they exchange this genetic material between the 2 homologous chromatids here. By doing that, basically you have generated a completely new chromosome here, which is a mix between the one from your mom and the one from your dad. This is referred to as crossing over, and practically speaking is done by a series of enzymes that what they do, they cut some of the DNA strand here. First one, they join it together through ligase, and they do the same thing with the other one. By doing that, basically whatever DNA is downstream from there get transferred to the other chromosome. Why is this important? Because basically this will lead, again, for example, this is your chromosome 1 from your mom, this is the one from your dad. These chromosomes, they have the same genes but different versions of them. Imagine this being the gene for the color of your eye, if it existed, just specific one. So this is for blue eyes, your same copy here. This is for brown eye. And now you have that some part of this DNA get exchanged. So now you have that this DNA here will start having a mixture of different alleles, so copy of the gene, which before did not exist. So you have generated a completely new chromosome through the process of crossing over. So summarizing, in prophase 1, the key thing, except for everything else that is similar to mitosis, you have that the homologous chromosome, they actually pair with each other, forming tetrads. The process is called synapsis, and this leads to exchange of genetic material by crossing over. The point in which the DNA gets exchanged, make contacts, is referred to as chiasma, the plural being chiasmata. Now, these tetrads, they don't get separated in the next phase. They still stay together until metaphase. So in prometaphase, what happens is exactly what happens in mitosis. You have that the mitotic spindle now has reached the two opposite poles, is fully visible, start making contact with the chromosomes. You remember the kinetochore microtubules? They make contact with the chromosomes. And then the nuclear membrane would have disappeared at this point. What happens next is in metaphase, you have that now the chromosomes here, they do align in the middle point, the equatorial plate or metaphase plate right here. If you remember, this is different than what happens in mitosis. So now you have that for each pair of chromosomes, homologous chromosome, one of them, whether it's being recombinated or not, would end up on one side or the other. So imagine this being the one from your mother, the one from your father for chromosome 2. The mother one end up on the top part, the other one on the bottom. And the same thing for these two. But this is completely random. You could have a different scenario. Just by random chance, you could have that for chromosome 2 versus chromosome 1, you have the one from your father on the top, the one from your mother on the bottom, and the opposite scenario for chromosome 2 here, which together with that already has recombination. So this random mixing or assortment of chromosome on one side of the metaphase plate versus the other is referred to as independent assortment. So this is completely unrelated. So these chromosomes, their combination will be completely independent from each other. Why is this important? Because these ultimately are the chromosomes that are going to be brought to one pole versus the other one. Now, if you remember also the big difference here, so differences again, pointing difference between mitosis and meiosis 1, or the metaphase and anaphase of meiosis 1, are in mitosis, all the chromosomes were aligned. So you would have that ultimately one of the sister chromatids for each chromosome will be brought to the two different poles. So these two poles will end up being, both of them, diploid. Because imagine this being your chromosome 2, the one from the mother and the one from the father. This cell now will get a copy from the mother and one from the father too. Same thing for chromosome 2 here. One sister chromatid from the mother, one from the father. So this cell would have a complete set of chromosomes, ultimately, both of them. In meiosis 1 instead, because of independent assortment and the homologous chromosome having one on one side, one of the other one, so this one, let's say, was from your mom, the one from your dad. Once these homologous chromosomes are pulled away, the cell, or the nucleus, generated by this first anaphase, anaphase 1, would have only one of the two chromosomes, two identical copies to sister chromatids of the same chromosome. So this one was from the mom, the one from the dad. So this cell will be, even though it has two copies of the same chromosome, but they're identical to each other, they count only as one. And so this cell we refer to already, or these two nuclei generated by anaphase 1 will be referred to as haploid nuclei already. And so this goes on, and so this is what we were saying before. So this is referred to as independent assortment here too, where you have that the chromosome here, the tetrads ultimately, they align in the metaphase plate here, and the orientation of the two chromosome part of the homologous pairs is completely random. It's independent from the other chromosomes. So you can have all possible combination here just generated by chance. Right here, you can see how this phenomena itself, just even in the absence of crossing over, will lead to 2 exponential 23rd possible combination. If you do the math, it's a huge number already, a possible combination, and therefore possible different gametes generated. by the independent assortment itself. So again, in meiosis 1, key steps are the formation of the tetrads in prophase 1 and the genetic mixing between different homologous chromosomes through crossing over. And then the second step is achieving metaphase 1, where the chromosome each chromosome of each homologous pair will go on one side or the other one, independently from the other chromosome or from the others. So generating all these independent assortment, and therefore generating all possible different combinations. Finally, telophase 1, so meiosis 1 ends with telophase 1, which is basically exactly the same thing that what happens in mitosis. You have now the one component for each chromosome. Homologous chromosome has been pulled on one pole. This will cause the formation of one nucleus and this another nucleus. So you would have the formation of two nuclei. Not in all cells, the nuclear membrane is going to be reformed. Sometimes it does, sometimes it doesn't. It depends on the organism. And there is a time in between, right after telophase 1, which is referred to as interkinases, which is that time that is basically between the first meiosis and the prophase 2 of the second meiosis. So once this happened now, you would have that the sister chromatids of each chromosome now that's been separated here will be further separated into two further poles there. So what happens here? Meiosis 2 takes place. Meiosis 2, just remember, is basically identical to what happened in mitosis, sorry. The only difference is that you're starting already from cells that have basically just one single copy for each chromosome pair. Two sister chromatids, but one single chromosome for each homologous pair. And so what happens in prophase 1, you already know, this chromosome, which the mitotic spindle is gonna move again, to the two opposite pole. So now it's moving in this direction. You would have one pole here, one pole here, one pole here, one pole here. That's in prophase 2. Now the chromosome, in prometaphase 2, they get contact with the new mitotic spindle here. And in metaphase 2, they get aligned in the middle, exactly like in mitosis in this case. They're all aligned in the mitotic plate here. And now, in anaphase 2, what happens? Exactly like in mitosis, one of the two sister chromatids get pulled one way, one in the other way. And that's exactly what is depicted here. So now you have the chromosome here, one sister chromatid has gone this way, one the other way for each of the chromosome present there. And ultimately in telophase or telophase 2, there's a mistake here, it's missing a 2. In telophase 2, as in mitosis, this is done, meiosis is done, the chromosome can start decondensing, relaxing, the mitotic spindle will disappear, and the nuclear membrane here start forming around each of these masses of chromosome, basically, that are being separated here. Then, in cytokinases, you would have that then this cell divides into 4, each one of them being a gamete with a single set of chromosome for each homologous pair. So 23 chromosomes each in humans in each of these cells right here. Now, this is an amazing process. I hope you get it. How much variability is causing by crossing over and independent assortment in meiosis 1. It has to be done in an extremely coordinated way. You can imagine how easily a chromatid or two chromatids, imagine just in this example here, you could have that both of them, these chromatids here, come back, imagine these chromatids don't really get separated, one this way, one the other one. They both end up on the same orientation, while the other they segregate well. You would end up with a gamete that has an extra copy of one of the chromosomes. So this can easily go wrong, and it goes wrong all the time in your gonads. Don't get scared, it's normal. And this is the base for what happens later on. So now you might have some gametes here and there that have more than one copy of each homologous chromosome. Imagine, for example, you might have 2 copies in one gamete of the chromosome 21, and all the other chromosomes that you have a single copy in the same gamete. Now, if that gamete fuses with a gamete of another person, which has a regular single set of homologous chromosomes, now you would end up having with a zygote which has 3 copies of the chromosome 21. And that's exactly how Down syndrome happens when you have a full chromosome 21. You have translocation too. Sometimes crossing over goes wrong. Crossing over might happen sometimes that one chromosome transferred one part to one of the cell, but it doesn't get that part back. And so you get one chromosome that's longer than the other. You would have a duplication. The other one would have a deletion. Many things can go wrong here. And sometimes, I'll show you later on, some things happen. So before we talk about that, we talked about always homologous chromosomes, and it's easy to understand this when it comes to the chromosome. They're the same length with each other. So chromosome 1, chromosome 2, chromosome 3, and so on. What happens to the sex chromosomes? Sex chromosomes are different from each other. You know how the base of sex is determined in males and females, in humans, but also in fruit flies. Human fruit flies, if you have 2 identical chromosomes referred to as X chromosome, you would end up being a female. Whereas if you have 1 X chromosome and a Y chromosome, as in us, myself, you would end up being a male. Now, these are different chromosomes lengthwise. So this picture here shows you the difference in length. So this is an X chromosome, this is a Y chromosome. These chromosomes do, when it comes to meiosis, they do have a homologous region which allows them to still form the tetrad as well. Okay, so to pair with each other and therefore being separated properly in the gametes. As I was saying before, process can go wrong, and there are some chemicals that unfortunately increase this rate. One example is BPA. I'm sure you're familiar with that. You always read it on plastic bottle, BPA free. BPA stands for bisphenol A. This is this chemical right here. And this chemical, what it does, it really interferes with the binding of the chromosome to the mitotic spindle. By doing that, so this will be, for example, your chromosome perfectly aligned, you're in metaphase in the absence of BPA. This is the same thing if you do it in a laboratory. You wait for the cells to align. In the presence of bisphenol A, you can see that the chromosomes in red here are all over the cell. Obviously, this will lead to the production of gametes if it comes to meiosis, which have all sorts of different numbers of chromosomes, which obviously will lead to incompatible zygotes, which are incompatible with life. One thing I forgot to tell you here, sorry, when I went through the example of Down syndrome, is that in reality, so there are these problems happening all the time. You have a lot of gametes which have all these odd numbers. However, you have to understand too that once these gametes, they fuse with an egg, most of the time, for most of the chromosomes, basically all chromosomes except for 2, they lead to trisomies that are incompatible with life. And they are the base of what we call spontaneous abortions most of the time. So where you have, there is fertilization, but the offspring that is produced, so the zygote and then the embryo that's produced, has one extra copy of one of the chromosome caused by defect in meiosis ultimately, and then these will lead to an embryo that cannot survive longer than a few weeks, maybe a month, and then suddenly will die on uterus. So that's a protective mechanism ultimately that we have in place to avoid obviously the generation of possibly a new species in every few fertilization process. All right, so we discussed everything we needed to discuss here. So in summary, what we talked about here is, first of all, the difference between asexual and sexual reproductions and the different ways that organisms deal with the different type of reproduction they have to produce variability, either through mutation in a sexual individual or through the meiotic process through sexual reproduction. Then we talked about how sexual reproduction takes place through meiosis, and then fertilization, meiosis leading to the production of gametes, so haploid cells, which then they fuse together through the process of fertilization, producing a new diploid individual. We also talked about, and I stressed quite a bit on, how meiosis introduced genetic variability through two main mechanisms taking place on meiosis 1, the first part of meiosis. This process being the crossing over, so which consists of genetic recombination between homologous chromosomes, which had formed the tetrads, and then the independent assortment of the homologous chromosome, which takes place still in meiosis 1, so in metaphase 1 of meiosis 1. And then we finally also discuss briefly what can go wrong, and also how additional fertilization, also there is additional variability that is introduced by fertilization depending on the gametes that ultimately get together. All right, I'm gonna stop here. If you have any questions, we'll have a meeting on Friday.